Fine day to everyone. Welcome to another Everything ALS Expert Talk series. Each one of us is part of this. Remember that. We are civilian-driven research, and that means that there's a place, if any of you would like to share any of your extra time with us, we have some wonderful research programs that we'd love to share with you. So if you're at all interested, you could contact us at info@everythingals.org. And with that, I'll turn it over to Casey.
Hi everyone, I'm happy to see all of you. Today our speaker is going to be Dr. Matthew Harms. Dr. Matthew Harms is an Associate Professor of Neurology. He received his medical doctorate from the University of California, San Francisco. Dr. Harms completed Neuromuscular Medicine and Clinical Neurophysiology Fellowships at Washington University in St. Louis. His clinical training in neuromuscular diseases led him into the laboratory of Dr. Robert Baloh, where his postdoc research identified the genes responsible for two orphan human diseases, dominant spinal muscular atrophy with lower extremity predominance and limb-girdle muscular dystrophy type 1D. He joined the Neuromuscular Medicine faculty at Washington University in 2009 with board certifications in neurology, clinical neurophysiology, and neuromuscular medicine. In 2011, Dr. Harms assumed leadership of the Washington University Neuromuscular Genetics Project and established his research lab to continue harnessing emergent genetic technologies to understand the causes of inherited neuromuscular diseases. His lab focuses on disease of the motor neuron, including ALS, and where his efforts have helped identify more than 5 novel disease genes. At Columbia, he continues these efforts, directing an international multi-site effort using whole genome and transcriptome sequencing to bring precision medicine to ALS. His lab efforts will occur in both the Motor Neuron Center and the Institute for Genomic Medicine. Please join me in welcoming our incredible speaker for tonight, Dr. Matthew Harms.
Well, it's a real pleasure to be here. Can everyone hear me?
Yes, perfectly.
Great. Then I will go ahead and share my screen. Hopefully you're looking at the full presentation instead of my notes.
Yes.
Anyway, it's a real privilege to be back speaking to the Everything ALS group. Just a little over 18 months ago, I came and gave a talk, and as I was reviewing that to make sure I didn't tell you the same thing twice in a row, I noticed that I was thinner and not nearly as gray just 18 months ago when I gave that talk. So if you go back and watch it, yes, I'm the same person. The thing in the background is the same for proof, but I do look a little different now. So I just want to begin by pointing out that I do receive active research funding from a couple of pharmaceutical companies and have received generous research funding from many over the years that have made lots of our understanding of ALS and genetics possible. And today, I'm going to focus on ALS genetics, what we've learned over the last 30 years, and then a little bit about what we're unlearning and why it's important that we have our hands open to information that we think we've acquired in the past, so that as new information comes in, we can refine our hypotheses and come to better clarity and more accurate answers. So let's begin by talking about the ALS timeline. So in 1869, ALS was first described by a neurologist by the name of Charcot in France. Of course, not much happened in terms of ALS research. In fact, most medical research for the next 100 years or so. And Lou Gehrig, of course, died of ALS in 1941. And it really wasn't until 1993 that the first ALS gene was discovered. And then very excitingly, and I think everyone on this call will join me in our enthusiasm for the fact that this year we saw the approval of tofersen for the treatment of ALS caused by the SOD1 gene. Across the top here, however, is arrayed all of the important landmarks in genetic discovery. So first, going all the way back to Gregor Mendel and the P's that we probably all learned about in elementary school or middle school. And the idea of heredity was first born really around the same time that ALS was first being described by Charcot. It wasn't until the dawn of the 20th century, though, that we understood that chromosomes were the material that were carrying our heredity. And it wasn't for another 50 years that we understood even the shape and structure of the DNA helix. Now, fast forward to when I was in elementary school and the first human disease genes were mapped. And then it wasn't until 2000 when we had really mapped most of the genes in the human genome. And then in 2010 was when it became possible for us to sequence the entire human genome for something approaching a research dollar. So this correlation between these two timelines, the ALS timeline and the genetic discovery timeline, brings us to an exciting parallel, I would say. And that is, I've now overlaid over this same timeline, year for year, the growth in ALS research publications. And so you can appreciate that as genetic discovery was kind of percolating along, as we were understanding DNA, really not much was happening in research, but correlating with exciting discoveries, including the first gene here, has been a dramatic increase in the number of publications and scientific discoveries related to the genetics of ALS. Many of the inflection points I'll point out in the next slide really parallel where new genes were discovered for ALS. And it's really genetic discoveries that have kept people's feet to the fire and kept the community moving forward for ALS discovery. So going all the way back to the very first ALS gene, which was discovered essentially 30 years ago this year, the SOD1 gene, two publications brought that about. One of them in Science and one of them in Nature, the two foremost journals in science. And these two groups, just months apart, described the SOD1 gene as the first genetic cause of ALS. Of course, since then, many additional genes have been reported in the literature to cause ALS. And really, if I squint my eyes at how these genes were discovered across time, I can pretty much hallucinate or maybe even actually identify kind of three phases, which I've highlighted in different shades of gray here. The first of these eras was driven by the existence of large families in which ALS had been passed down to multiple generations. Those large families with DNA available allowed for a very powerful technique called linkage analysis to identify the exact region of the human genome that was responsible. And then it would take really the combined efforts of multiple postdocs, entire careers, to map and then identify what gene was within that region of the genome. And so you can appreciate that basically from 1993 to 2008, about one gene, a new gene was reported, less than one gene was reported every single year. Then in 2008, the ability to sequence the entire genome at one time instead of having to look at just snippets of the genome became possible. And so this is what I call era 2. Here, we took advantage of smaller families. Most of the large families had been solved during era 1. But you could combine multiple families, multiple small families, and do whole genome sequencing. And during this era, 3 genes were discovered per year. So you can see a tremendous increase in the slope of gene discovery pace. And now I would argue that we are in what I call era 3, which is where we're essentially out of large families that powered era 1 and 2. And now we need to turn our attention to patients who have so-called sporadic ALS with no family history. And we're able to do that because whole genome sequencing has gotten very cheap. Because of internet technologies, we've been able to connect groups across the world to combine forces. And that has given us statistical power that has allowed us to approach these whole genomes from sporadic ALS patients to identify snippets of the genome. And so now I would estimate that we're still identifying and reporting 2 new ALS genes per year, which is tremendous progress, and I hope you'll agree with that. So if you're curious for more detail about the methods by which we identify these genes in any of the 3 eras that I've just talked about, or specific details about some of the specific genes on this list, or even asking questions like, what's the human genome? Or how do mutations in individual genes lead to ALS? I'd like to refer you to that talk where the younger, thinner Matt Harms gave a talk on the genomics and genetics of ALS. That is on the Everything ALS YouTube channel. And I cover some of the basics about the human genome, how gene mutations give rise to mutated proteins that don't do their job. And then importantly, how we think genetic therapies that are being developed can tackle each of those different causes when things go wrong. There are also, I was looking today, some really great talks recently by Jonathan Kupernak, a colleague and collaborator of mine, about how genes interact with the environment, potentially to trigger or contribute to the progression of ALS. There's an excellent talk by Dr. Bob Brown covering the exciting developments in gene-based therapies for ALS. And then finally, some excellent talks by several genetic counselors, including Elizabeth Harrington here at Columbia, talking about genetic testing and genetic counseling for ALS. So I showed you that slide of all of those genes that have been discovered over the years and reported to cause ALS. And I just want to briefly highlight just how common they are, so you have that for a frame of reference. So here I've taken definitive ALS genes, and we've analyzed them now in 5,600 patients with ALS. And I'll point out one important fact, and that is that for years and years, most of the DNA collections from patients living with ALS took place in European and North American countries. And so we have the richest data coming from people of European ancestry. But thankfully, much attention is now turned to non-European populations. And so there are efforts in India, China, other parts of Asia, even some countries in Africa and South America to really go after collecting DNA samples so that we can have more equitable understanding of the distribution of these disease genes across all geoancestries, across all continents, and not just coming from predominantly white populations in Europe and North America. So when these definitive genes are sequenced in 56,000 people with ALS, the breakdown is essentially like this diagram. The vast majority of genetic abnormalities are in the C9orf72 gene, followed by the SOD1 gene, followed by TDP43, and then they get rarer and rarer from there. If you total it all up, we expect that around 11.5% or so of people with ALS will have an identifiable mutation in the genes that we know about today. And a fraction of those, around 3.5%, are in non-C9orf72 genes. And I've shown some of the most common ones from this particular analysis here. I will point out that we already know from studies of relatively small cohorts coming from non-European countries that outside of white populations, there's a much lower rate of the C9orf72 gene, but higher mutation rate in the TAR-DBP gene and the FUS gene. And I go into a little bit more detail on that aspect of things in that other talk that you can go back and take a look at. Excitingly, a lot of therapy development has been taking place around these particular genes. So as many of you know, we now have an FDA-approved antisense oligonucleotide called tofersen, or Qalsody, that's been approved for patients with causative mutations in the SOD1 gene. There are clinical trials ongoing or recently completed for C9orf72, the FUS gene, and the ataxin-2 gene. And then with permission from the FDA, we're even developing antisense oligonucleotides for rarer and rarer gene mutations in what we're calling a precision medicine or N of 1 initiative called Silence ALS, where we're developing antisense oligonucleotides that target one or a handful of specific mutations in some of these more rare ALS genes. I won't talk much about that, but I hope you share my enthusiasm for the fact that things are moving forward for the treatment of genetic ALS. And I'll just throw in a little plug here because I won't have time to talk about it. But some of these same technologies, these antisense oligonucleotides, may play a role in treating cases where we don't identify an ALS mutation by modifying some of the shared features and the shared places where all of these genes are impinging upon one another, which I'm very much looking forward to. So the fact that we find genetic mutations in almost 12% of people with ALS, and the fact that we have clinical trials for many of the genes and an approved medication for one of them, led some colleagues and myself to push for developing testing guidelines for ALS genetic testing. And I can summarize the outcome of those, which have now been accepted for publication in a journal and should come out very shortly, is that we're recommending, and the evidence supports us recommending, that we should test everything in everyone all at once. What do I mean by that? I mean that lots of people have an identifiable mutation, and we can't tell, even based on family history, whether someone will have a gene or not. And so we shouldn't be guiding our testing based on family history. We should be offering genetic testing to everyone with ALS because of the high likelihood of finding a genetic mutation. That maybe didn't matter as much before genetic therapies were available, but now that they are, it becomes doubly important. We also feel like everything, like all of the genes on the panel, should be tested at the same time, because we are increasingly recognizing that there are people with who are what we would consider dual mutation carriers, who have more than one abnormality that may be contributing to the disease that would be important to know about. And then most importantly is the fact that if we've sequenced everything all at once, we don't have to go back and keep sequencing additional genes, which will shorten the time to receiving a treatment or to getting into a clinical trial if a genetic mutation is ultimately found. So for those of you on this call, if you've attended before, I'm sure you probably are aware that genetic testing is relatively widely and cheaply available here in the United States. It's not quite as easy to obtain or always free in other countries. But there are two programs, one at Invitae and one at Prevention, where genetic testing can be obtained by anyone with ALS for free. So when that's done, just to illustrate again what we expect to find, I'd like to just show you 100 people living with ALS. In this first row here, approximately 10% will have a positive family history of ALS. But that means that the rest of the group, the other 90%, are what we will consider Singleton or sporadic ALS. You've probably heard it called sporadic ALS. Geneticists prefer the term Singleton because it conveys that there's one person in the family, whereas sporadic makes it sound like it just happened out of the blue, that it was just a freak chance, when in fact we now know that there are some genes that can be contributing. So if you now take a look at those with the family history of ALS and ask what genes are identified, this is the breakdown. 4 out of those 100 people will have the C9orf72 gene, 2 will have SOD1, and then an additional individual will have one of the rarer mutations. If you now do all that testing in individuals with sporadic disease, you'll find more individuals carrying C9. You'll find some more with SOD1 and some of the rarer genes. And I think you'll appreciate that we actually find more individuals with gene mutations from the Singleton pool, from the sporadic ALS patients, than we did from those with the family history. So what does this tell me? This tells me that there are more genes that remain to be discovered, even though we've made significant progress. So just to remind you of all of the ALS genes that have been reported, and I just highlight that the reason why we need to continue looking for new ALS genes is because with each gene we identify, we hone in on different pathways in a cell or different mechanisms that might lead to ALS. So for example, when we identified senataxin as an ALS gene, that told us that the ability to wind and unwind DNA was important in ALS, and we now understand that DNA damage plays an important role in ALS. When we identified TAR-DBP or TDP43, that told us that RNA metabolism and the regulation of splicing was important. Ubiquitin 2 told us that autophagy was important, and that was emphasized by profilin 1. And so you can kind of see that as we identify each of these genes, we learn more about the different pathways that are important for ALS. And I like to think of it kind of like this diagram here of the world's tallest climbing wall, that each of these genes is essentially one of these handholds that we're climbing up this really tall wall, and eventually we'll get to the top using each of these handholds. So we're using these handholds to inform what we call driver mechanisms. So if you look at all of the genes that have been discovered for ALS and put them into different categories, you can start to appreciate that, hey, there are a half a dozen or a dozen genes that implicate a process called protein homeostasis, how proteins are folded, where they end up in the cell, how they get recycled. Another almost dozen implicate something called RNA metabolism, which is about how genes get turned on, how they get utilized, how much of the gene gets turned on, how the gene gets rearranged and utilized. We also find that the ability to send things from the cell body all the way out the axon of each neuron is important, and cytoskeletal and axonal transport plays a role there. And so we learn from each of these different DNA discoveries about the fundamental cellular pathways that are important to ALS. And so not only are there more genes to be discovered, but there are more pathways to be discovered. I'll just talk briefly about how we're going about that, because I hope it will bring some optimism that we're on the cusp of some major additional gene discoveries. So when I first started sequencing the genomes of patients with ALS, we carried out what was called the ALS Exome Sequencing Consortium. This was one of the first studies to bring together DNA samples from all over the world. People were willing to contribute those because we were going to give them free sequencing. So they would find out about all the different gene mutations in their particular patients. And when we did that with 9,000 participants, we were able to identify two brand new genes, TBK1 and the NEK1 gene. We've kept going. In our next iteration, we were able to increase that to 11,000 participants. And not only did we re-identify SOD1, meaning that if we hadn't known that that was already an ALS gene, we would have rediscovered it. We also were able to identify TDP43 and FUS, and then implicate several more interesting genes that we're continuing to take a look at, including one called PKP4. That was all the way back in 2019. Our current iteration has expanded to 27,000 participants, and I won't talk much about that because we're still in the process of writing up the results. But encouragingly, I would say, is that just around the corner, literally within the next year, is another iteration with more than 100,000 participants from around the world in a project that's being led out of the National Institute of Health and UMass Worcester, called ALS Compute, where all of us are combining our samples and our data, bringing in information from a European equivalent called Project Mine in order to look at more than 100,000 people. And we really expect exciting additional gene discoveries coming from this many individuals. So this is going to lead to the discovery of new ALS genes, but I already showed you this diagram with many genes that have been identified and reported in the past. And you probably noticed that there was some color coding that I hadn't given any time to describing so far. And I'll do that now by basically saying that when a group of us went back and looked at these reports over the years, we were able to find that many genes had what we called ample replication data, meaning multiple groups had found the same answer when looking. And those you can kind of take to the bank as definitive ALS genes. Many others, the ones that I've colored here in yellow, had some replication data, or maybe had been studied in cells, and that was consistent with potentially causing ALS genes or causing ALS. But then there were quite a few where there was no replication data. Mainly, one study had identified this gene, or there was very minimal experimental data to support causation in ALS. or where ALS was kind of a rare manifestation and usually it presented as a different disease. And we thought it was important to get the word out there that the strength of evidence for specific ALS genes varies from gene to gene. And it was important to take a look at that systematically so that everyone was on the same page in terms of which genes truly cause ALS and which ones might not. This has several important implications that I'll talk about shortly. But I want to make the argument that this has to happen as a community. It has to happen in a systematic fashion, and there are important reasons for that. The first is we're not the only people to have noticed that the strength of evidence for different disease genes varies. This is happening across all of human genetics, not just in ALS. This recognition that we have to go back and come up with a standardized way to classify these different genes. And even within ALS, people had taken a stab at it. So ALS online database run out of the UK, which is extremely reputable and has been around for more than a dozen years, has its own ranking system: definitive, strong evidence, moderate evidence, tenuous, and so forth. But this is really brought about by kind of two experts reading the literature. Genomics England has crowdsourced experts and kind of gives a green, yes, it causes the disease, red, we're not exactly sure, it's insufficient. And then there's the several expert one that from the New York Genome Center and ourselves that I just referenced. And everybody's coming at this from completely different directions. And so an organization called ClinGen has created a framework to bring experts from all over the world together to help bring about this clarity. Not just for which genes cause disease, but kind of across genomics and genetics. And so I won't go into most of these, but they have multiple different routes to clarifying and refining the information that we have for diseases and genes. Essentially, here's how it works. So basically, you identify a gene. Someone goes through and does an extensive literature search to find all of the scientific data that supports a particular gene. We go into a database called the ClinGen Gene Curation Interface, and somebody manually enters hundreds of data points, basically, for each of these genes. Then they come up with a classification selection based on that data. That gets reviewed by a panel of experts and then gets approved for the entire community to use. And there are several things that I really like about this. So one is that it's quantitative. So there are scores that get issued for each of the different categories. There are scores for the number of families. There are scores for the different variety of the mutations that are reported. There are scores for whether animal models exist, or whether cells have been looked at, or whether autopsies have been looked at. And each of these gets a score that very accurately predicts, based on studies that have been done, whether a gene is actually causative of the disease that it was reported for. So once all that's done, our committee meets and it generates a final voting recommendation that's available to anyone online. So some of those other organizations, including the data that I pulled together and showed you at the beginning, is far from transparent. We each came up with our own criteria. That's not necessarily published. But here it's completely transparent. The scores that were given and why those scores were given, the papers that underlie those scores are all available for people to look at, as well as a synopsis that's predominantly for clinicians and for people who perform clinical genetic testing to be able to take a look at. All available online, and that goes live almost essentially as soon as the decision has been made for individual genes. There's also a talkback option, where basically when you look at it, you can give feedback on the evidence. So any expert who takes a look at what our committee came up with can dispute or bring to light new information that maybe was overlooked the first time around to be considered in a reevaluation of this specific gene. So here is a fraction of the people who have participated on our panel. We have considerable geographic diversity with 19 experts spanning 8 countries on 5 continents. We have genetic counselors, people who run labs devoted to informatics, basic science of ALS, and then 11 clinicians who primarily focus on ALS and genetics. And we've now been working for almost 3 years. We've done more than 800 hours of curation activity, of reading the literature. We've had more than 52 conference calls to discuss what is being presented. And that's resulted in more than 650 person hours of deliberation. So lots of work and effort have gone into these, I would argue, highly reliable gene classifications. And we've gotten through 39 of the 52 genes that we plan to tackle. And this is the breakdown as to what the evidence shows. So 45% of the genes that we've reviewed so far have meet the definitive criteria for being a certifiable causative gene for ALS. And I've listed a handful of those genes here off to the left. A smaller number are almost to definitive, but could use some additional either animal model data or scientific evidence to support them actually causing ALS. But almost an equal number have fallen into this limited category, where basically, although the initial papers may have claimed that they cause ALS, familial, genetic ALS, the data just isn't there. And that group includes some very famous ALS genes that have been sequenced and talked about for decades now. And then finally, even a handful of genes which are what we have termed refuted, meaning there's no good data that they don't cause ALS. So it could emerge in the future as we do additional studies that some of these genes do play a role in ALS. But these, we've pretty much seen enough evidence or data now that they don't contribute to ALS. And yet, as I'll show you shortly, most of these refuted genes continue to be sequenced and are on ALS gene sequencing panels at sequencing laboratories around the world. So these genes are for sure false positives from our discovery efforts over the last 30 years. Some of these may turn out to be false positives. Some may, with more evidence, move up into the moderate category, and some of the moderate could move into the definitive category. But I suppose in the mind of a patient, that raises the question, how could there be false positives in the first place? Genes that people claimed cause ALS, but most likely do not. Well, let me walk you through how a typical study design happened in the past. So it would go something like this. A scientist would think, hey, TAR, DBP, and FUS are definitely causative for ALS. And we know that those are RNA binding proteins. So I'll bet there are some other RNA binding proteins that could cause ALS. So I'm going to sequence those in ALS patients. And because of the cost, they would sequence 20, 25 RNA binding proteins in 500 ALS patients. And then all of the mutations that they identified, they'd go back and look in 100 controls. And all of the ones that they didn't find would be assumed to be causing ALS. And so they would come to the conclusion that these new RNA binding proteins cause ALS. So we now know that there are several flaws and problems with that thinking. First, it's biased. And that's not any knock against the researchers who did it this way. At the time, that was of necessity. We couldn't sequence the whole genome. It was just too costly. The studies were too small. That was also of necessity because of cost and because it was hard to find patients with ALS back in the 90s and 2000s. You couldn't just put out a call in clinics or bring together people from all over the world because the internet barely functioned. And then finally, I think most importantly, is that we didn't yet recognize the enormous diversity of genetic variation. And that really within each of us, there are somewhere between 20 and 30 completely unique genetic variations that have arisen just in us. And so we didn't realize just how complicated that would make these types of analyses. So that's how false positives ended up in ALS gene lists. Actually, ALS did better than many other fields. More than 25% of cardiothoracic cardiac genes end up in that category. So we're actually doing better as a field. But there still are these false positive genes, and these genes actually end up on sequencing panels. So the same committee I was telling you about surveyed the testing panels that are most used around the world. We identified 14 panels, and actually 91 unique genes were sequenced on those panels. Surprisingly, 44% of those were only on a single panel, which means They were either missing, if they're a true ALS gene, on other panels, or they were fluff on panels that were including them because they're not actually known ALS genes. And I think this argues very strongly that we need to come to a consensus about which genes are truly ALS genes so that our sequencing panels can be refined to include just those. 20% of panel results, so if you have genetic sequencing, result in a variant of uncertain significance, and most of those occur in those genes that have limited data. And so if we paid close attention to which genes should be sequenced, which ones we have the great data for causing ALS, then we would eliminate many of the variants of uncertain significance. So why should we eliminate these false positive genes? Why should we spend any time looking at them? I know we were talking about positivity at the beginning. This kind of seems negative, but here are some positive reasons to eliminate these false positive genes. So the first is that this will help us to eliminate anxiety during genetic testing. So the recommendations from our committee have been that we stop testing and reporting variants in genes that have limited data that they cause ALS or have been refuted. That makes a little bit of common sense. And then for laboratories that are unwilling to remove those genes from their testing panels, that they actually make it clear in the report what the level of evidence is for the gene that they're reporting. So if they find a mutation in a gene called DAO, which falls into the limited evidence of our panel, that the report would actually say, although there was a variant found in DAO, the ClinGen gene curation expert panel has found only limited evidence that this gene is causative for ALS, so that clinicians would have the opportunity to take that into consideration and help families to understand that. A second reason to eliminate these false positive genes is that it will allow us to reprioritize ALS research spending. So we would be able to stop studying disputed or refuted ALS genes, that 5% that we don't think cause ALS, but people continue to study. And it would allow us to focus on changing those genes with limited evidence from studying their mechanisms, like kind of assuming that they already cause ALS and jumping straight to trying to figure out how they cause ALS, and actually use those research dollars to clarify whether they are truly causative of ALS. And then I think finally, the strongest reason to eliminate these false positive genes is because right now people are focusing heavily on what I call shared mechanism research. So shared mechanism research, you basically make a neuronal cell line from somebody with a mutation in gene X and a mutation in gene Y and somebody with one from gene Z. And then you look at some shared feature, maybe it's all of the genes that are turned on or upregulated, and you overlap them. And so you would say, I'm interested in this shared mechanism between these three genes. And if you kind of look at the intersection of these Venn diagrams, you would then naturally focus on these 10 genes where all three gene mutations led to an increase in the gene expression. But what if gene Y is actually one of those false positives, and you've included it in this analysis? You would be focusing on those 10 there in the center of the diagram. But if that one were eliminated and you weren't considering it, then you would actually be focusing on these 40. And so if the actual key to ALS, that mystery thing that McFinn was talking about before we started in, that hopefully we'll wake up one morning and find in the paper as having solved ALS, could have been over in these 30 genes that you were going to ignore while you were still considering gene Y as a true causative gene for ALS. And so that's why we're working so hard at this, because it's essentially free to eliminate false positive genes, whereas it's pretty expensive to go out and discover new ones. So while we're working on both of those things, it just makes sense for us to eliminate these false positive genes. We also want to take some time to eliminate variants within those genes, specific mutations in those genes that may not be causative. So let me now turn for the remaining of my time to variant interpretation or variant interpretability. So I mentioned earlier that we didn't really know just how vast the genetic diversity was amongst humans until maybe 10 years ago. And so as you look at all of the variants that have been reported in human disease genes, they've just massively exploded over the course of the last decade. That's also true for ALS. I'm just showing these top most common ALS genes, SOD1, TDP43, and FUS. And you can appreciate that over time, the number of variants that have been observed in humans, people with ALS, people without ALS, have massively expanded. And it's really important for us to get at which of the mutations that have been found in these genes actually cause ALS and which ones don't. And that's an essential fact and something that has to happen if we're going to understand why these genes cause ALS and bring precision therapies to ALS genes. So here I want to just zoom in. That we have a way of ranking the different mutations in ALS genes as to whether we think they cause ALS or not. So now these are genes that we're definitively sure cause ALS. We still don't know how to, we're still working out which genes cause ALS, but these are the genes we know cause ALS. And now we're asking the question, which specific mutations in those genes are capable of causing ALS? And so we have pathogenic or likely pathogenic, which is an estimation by some criteria that the mutation is likely to cause disease. We have these ones in green, which we call likely benign or benign, which are ones that we're pretty confident don't cause ALS, even if you have one. And then there's this other category called uncertain significance, which are the ones where we're just not sure. They don't fall into either the pathogenic, likely pathogenic, or the likely benign or benign category. And it's essential that we actually, first of all, double check to make sure that everything in red, pathogenic and likely pathogenic, is truly causative of ALS. That same false positive problem that I mentioned when we're talking about genes also applies to gene mutations. We also need to make sure that what people have called benign are truly benign and actually don't increase the risk of ALS. And then equally importantly is for us to bring some clarity to these variants of uncertain significance. In some cases, the fastest enlarging group of gene mutations. So how do we do that? Well, we've gone back to that same committee, the ALS Gene Curation experts, and have asked them to form a variant pathogenicity committee, also called a VSEP for Variant Curation Expert Panel. And we're now going to go through all of the variants and apply standardized metrics to figure out which ones truly cause ALS and which ones don't. So why would we do that? Hopefully you'll appreciate that this is essential for providing accurate genetic information to patients and their families. I should point out that there are some families that have been told they have ALS caused by the angiogenin gene or the ANG gene, or caused by the FIG4 gene, both of which are now in that category of very limited data that those genes even cause ALS. And then there are also families who have heard that a specific mutation in a specific gene is the cause of their ALS, and that needs to be reevaluated to make sure that that in fact is true. So we want to be able to provide accurate genetic information to patients and their families. We also need it to guide the appropriate use of these new round of genetic therapies that are becoming available. So let me give an example for tofersen. Tofersen is the antisense oligonucleotide that treats ALS caused by SOD1 mutations. But in the very first year of treatment, it requires 14 spinal taps. Each of those requires the time commitment on behalf of a patient of at least a half a day to come to clinic and to get dosed, monitored, and then get home. Each of those spinal taps has the risk of a post-spinal tap headache. And it brings with it a very low risk of increased pressure on the brain, the optic nerves, or nerve inflammation. And then, of course, that's not even considering the cost. And people may have very high copays for both receiving the drug as well as the medication. And so if somebody, in fact, has a gene mutation that's causative, they have a chance of benefiting that will offset those risks and offset the cost and the time commitment. But if they have a mutation that isn't truly causative of ALS, then they only stand to incur these risks, costs, and commitments without any chance of benefiting. And so it's super important that we identify not only which gene mutations do cause ALS, but which ones don't, so that we can fine-tune the use of these genetic therapies. And then I think lastly, and I'm getting close to the end here, is we need to clarify which variants cause ALS to guide those same comparative mechanistic studies that I was showing with that Venn diagram. So it's the fact that for most ALS genes, we don't yet know for sure how causative mutations are acting. And so we need to plow a lot of research dollars and effort into figuring that out. And in order to do that, we need what I would call gold standard causative mutations and gold standard benign mutations in order to do comparisons. And so if you knew 3 causative mutations and 3 benign mutations and tested them in some paradigm, and you then saw that all of the causative ones were giving values of 15 to 20, but all of the benign ones, the ones that don't cause ALS, were less than 5, then you would be onto something. Like this would be giving you a clue as to how the causative mutations were in fact causing ALS. And of course, the eventual goal would be that if you then identified 3 new mutations in that gene, variant G, E, and F, you could then put them through that same test. And you'd be able to say, oh, hey, G definitely is a cause of ALS because look, it's behaving just like the causative ones. E, however, is likely not causative because it's acting like all of the benign ones. And then we would be left trying to sort out what it means if you got an intermediate result, like variant F here, where it's kind of in between the two. And then that would be really spur us on to additional research in important ways. So we've now created this amyotrophic lateral sclerosis variant curation expert panel. We're aggregating information from companies, from clinicians, from a ClinVar database and ALSOD, and we're going to standardize, do literature review, pull in unpublished genetic data, unpublished experimental data, and come up with classifications for gene mutations in specific known definitive ALS genes. Sorry about the arrows there. And this is going to allow us to resolve variants of uncertain significance using all available information. And it's going to ensure that we deploy those genetic medicines to appropriate mutations. This is the committee as it stood a month ago. We've now doubled in size. And I want you just to appreciate the sheer scope of what faces us. So here's the SOD1 gene. These are all of the mutations that we're going to take a look at, each one of these little tiny squares. For the FUS gene, these are the ones we're going to tackle. And then for the TDP43 gene, these are the ones we're going to tackle. And for sheer scope, that means we're going to be looking at a great level of detail, more than 750 mutations in just these 3 genes alone before getting to the other genes in the database. So if you thought the number of hours of curation and the number of hours of meetings that I showed you for the gene curation committee were huge, you can kind of imagine what it's going to look like for this. And so we've asked the National Institutes of Health to fund a grant to cover this so that we could actually hire people to make this process go faster. And we've also approached individual nonprofits to consider providing grant funding to this committee as well to enable this process to go as quickly as possible. So I've got one conclusion slide. The last 30 years of ALS gene discoveries have been absolutely revolutionary. We've discovered quite a remarkable number of genes that cause ALS, and through those have learned about many pathways that are essential to causing the disease. And I would argue that additional gene discovery needs to take place and is already taking place with discoveries that are soon to come, I would predict, in the next year or two. But I would then also argue that it's important that we continually reevaluate the evidence for what we thought we knew already, both at the level of the genes and at the level of the variants, to really refine our hypotheses and help us to guide and reprioritize ALS research funding around the places that are most likely to yield definitive therapies for people living with ALS. And I'll go ahead and pause there and turn it back over to the hosts to fire away with the questions that you all have.
Thank you, Dr. Harms, for that very informative presentation and for all the work you do. Brian and I will start the questions now. The first one is, how do you give names and numbers to the genes? Is there any method for this?
So the names come from an organization called Hugo, which has basically established criteria. If you find a gene in a fruit fly, you get to name it something clever. There's even one that influences how much alcohol it takes to get a fly drunk called cheap date. Funny things like that. But in humans, it needs to be named after a function. And so most of the genes are named after the function that we know about at the time of discovery, though that might not be the function by which ALS is caused. But there's a nomenclature organization that decides.
Perfect. Next question is, can you expand on DNA damage and its role in ALS?
Yeah, this is an emerging topic. We now know, one of the questions is the DNA damage that we see in ALS part of the cause of it, or is it the result of ALS? And I think that's still being worked out in many circumstances. But if you look in C9 cells, you find far more DNA damage than in normal neurons. And that, I think, is kind of true across many of the different ALS genes. How that DNA damage occurs is still a matter of active investigation. And, it's challenging to think about how you repair DNA once it's damaged. And so I think the treatment strategy that will kind of emerge for that will be to try to prevent it in the first place. Because once the DNA is kind of chopped up or chewed up or sustained additional mutations, it might be too late. So we definitely want to focus on preventing DNA damage, whatever its cause happens to be.
This is a follow-up to the previous question, but a participant asked that her sister was diagnosed with juvenile rheumatoid arthritis as a child and was given lots of different types of medicine, including gold injection. And she was wondering if this could have harmed her DNA, leading to her ALS diagnosis 40 years later.
Almost impossible to know. The number of people who were treated with gold a very long time ago was high. And I don't know of any studies that have, shown an association between previous gold therapy and the development of ALS, but it might be too rare to have been able to see that type of a signal. But, we're still trying to understand all of the different factors that could contribute. And for sure, DNA damage from many different sources could be a contributing factor.
Perfect. So next question is, how many genes are typically screened for within current testing? And for a follow-up, should a patient be tested yearly as new genes are being discovered?
Let me start with that one. So companies add ALS genes more carefully now. So our genetic testing guidelines, I think, will spur that further, so that people will wait until the evidence that a gene causes ALS is pretty robust before adding it to a testing panel. So that means that testing panels don't get revised very often, maybe every 5 years. So getting retested, wouldn't make sense, because it's essentially going to be the same gene panel that you got previously. Where most people are moving is to skipping testing panels altogether and to testing the whole genome. So that basically on a thumb drive, you have a list of all of the places where your genome is different from the average human. And then as new genes get discovered, somebody can just take a look on your thumb drive to see if you have a mutation in that new gene, so you don't ever have to go back and get resequenced. And I think that's probably the way we'll be doing things, within the next 5 years. I saw somebody ask specifically about the time, 4 years ago. It depends which panel was used. Some companies have updated their panel since then. Others haven't. And so you could talk with your clinician about whether enough new genes have been identified, or which panel was used. Sometimes the panel 4 years ago didn't include the C9orf72 gene. So kind of having a detailed understanding of what was on the panel and what wasn't on the panel would be helpful in knowing whether you need to get retested.
How do scientists know that they are finding gene mutations that are involved with ALS versus just gene mutations that are present in people with ALS but not necessarily involved with the disease? And then how do you think AI will change the gene discovery process?
So So the way that we're finding new ALS genes, and I would argue that the way we're doing it now is finding true positives, not false positives. And the reason for that is because we're using statistics to identify that. So statistics require large numbers of individuals participating, and that's why I was saying that our next iteration of gene testing with 100,000 participants will be more powerful than the one that we've completed with 27,000. So we can use statistics to basically find regions of the genome, specific genes in the genome, that are just so astronomically overrepresented in ALS patients that there's basically a 1 in a million chance that we're wrong about it causing ALS. And so the statistics are really what's enabling us to identify new ALS genes. Now, the way we identify those is by something called burden testing. And burden testing requires us to define a DNA variation that we think causes disease versus a variation that we don't think causes disease, and then to basically add them up. And the way AI is influencing the way we do these studies is by helping refine those definitions. And so you can take everything we know about known ALS genes and all of those gold standard mutations that I talked about our committee coming up with, feed those into an AI machine and ask it to look for characteristics and themes that the human eye and the human brain couldn't discern. And so there are companies that have used this approach to try to figure out which mutations are more likely to be causative versus which ones are more likely to be benign. And then you can feed that into that big statistical model to hopefully power more rigorous identification of ALS genes. There are other ways that AI is going to get involved in the future, but that's one way that it's already impacting how we are doing these types of studies.
Perfect. So next question is, so we have a participant who's from a family of with ALS, and then their question is, how can they obtain genetic counseling? And once they obtain genetic counseling, what will the results be from genetic testing?
So, it's hard to predict what the results will be. I would say the most likely thing is that your test would come back negative. That happens 75% of the time. You do the whole panel and the test says we didn't find anything that wasn't expected, in the background of human variation. The next most likely thing is that a variant of uncertain significance will be identified. That means an abnormality in a gene that we don't know how to interpret today. And those are the ones where genetic counseling is extraordinarily helpful to kind of help understand kind of why don't we know more about it, or what's in the way, or what do we know about it. And then finally, the least common outcome is to find a gene mutation that we know about, we understand, and is definitively involved in ALS. And that happens somewhere between 10 to 12% of the time. Getting genetic counseling can be a challenge. Genetic counselors are expensive, and Medicare doesn't pay for genetic counseling. And so it can be a challenge to find it. Some of the free genetic testing programs offer free genetic counseling after the fact. So if you have a positive or a variant of uncertain significance, if the person who ordered the test checks a box saying the person's interested in genetic counseling, then that can be obtained for free through the genetic testing company. And I would highly recommend getting genetic counseling. It can be really confusing to try to understand the different things that pop up on genetic testing.
How do we confirm that something is a dual mutation, since usually dual mutations are inconclusive?
Right. So that is true. They usually are inconclusive, which one might be the culprit. But as we sequence more people, that information is going to come to light. And what I would hate to have happen is for someone not to know about their second potential culprit because they only looked at one of the genes. And so they found the SOD1 mutation, but they didn't see the rare variant in the TBK1 gene, for example. And in 5 years, we're going to know that those types of combinations behave differently than either one of them alone or something like that. So it's a little bit what we call future proofing, so that basically the information is in your chart, it's in somebody's hands when we catch up with our knowledge base. So that question is exactly spot on. it is difficult to interpret at this moment, but will become easier as we get more information with time.
Perfect. So next question is, once a causative gene is identified, is preventive treatment possible before ALS appears or ALS symptoms, etc.?
Yeah, so we sure hope so. We don't know for certain. That's a hypothesis that if we get in early enough with genetic therapies, you could prevent ALS. And the ATLAS trial being run by Biogen is asking whether giving tofersen shortly before we expect symptoms to come on can delay symptoms from coming on. Many of us are working very hard to identify every pre-symptomatic or asymptomatic gene mutation carrier in the country so that we have a database so that basically as soon as we know that something like that is going to prevent ALS, we can get it into people who will stand to benefit. And then we're also working hard to identify additional antisense oligonucleotides that would be applicable to other genes, even at the time of symptom onset. So for example, here at Columbia, Dr. Schneider, my boss, and I are working on this program called Silence ALS, where we develop these antisense oligonucleotides that are predicted to be applicable to one family mutation. Maybe there are multiple families around the world that have those mutations, but one family mutation. And so we know the person who has the disease today, and that's the person we want to treat up front with the antisense oligo as soon as it's available and possible. But then we want to identify everyone in the family who has the mutation, who may develop symptoms in the future, and be able to get that antisense oligonucleotide in preventatively. Though there's lots of steps, and the FDA has to come along, and there's many hurdles to overcome in that. But that's kind of the ultimate goal, would be to prevent any neurons from deteriorating from the ALS disease process.
Okay. Thank you again, Dr. Harms, for joining us tonight. This will be our last question before I hand it back to McFinn. Can you please explain how treatment for fused genes works? And does each gene have only one function? For example, if a gene is found to cause ALS, what else is that gene causing?
Sure. So the last part is easy. Genes definitely have more than one function. And so you have to be cautious if you're going to take an approach like an antisense oligonucleotide, because it will delete the gene or prevent, it doesn't delete the gene, it decreases the gene product being made from the gene. And if it has another function you didn't know about, then of course that could be a side effect, right? You could turn off a gene that's essential. So before proceeding with an investigation with an antisense oligonucleotide to decrease a gene expression, you want to know as much as possible about its function, and you want to know when it's important. So for example, the FUS gene plays an important role in all sorts of things during development, but then we think it shifts function and maintains motor neuron health in adulthood. And so you could decrease it in adulthood without causing developmental problems, because development has already happened, for example. But it is important to understand as much as possible about a gene before decreasing it with an antisense oligo or with any type of mechanism, because you could run into what we call off-target effects. So side effects that come from inadvertently suppressing the function of a gene that you weren't expecting. So to answer the first part of the question, the antisense oligonucleotide ulefnersen, which is in the clinical trial called Fusion, run by Ionis, works to decrease the expression of the FUS protein, and thereby hopefully decrease the mislocalization and clumping up of the FUS protein that we think underlies part of the disease.
Okay, I think that is our last question for tonight, McFinn.
All right.